Hello and welcome to the Polymath World channel, and we're talking more genetics today, which you can't blame me because I love it so much. And I'm here with a friend who I met several years ago at the Genetic Society conference on the 200th anniversary of Gregor Mendel's birth. So it seems very fitting today that we're talking about plant genetics. I'm here with someone who really inspired my students as well. I'm here with Dr. Lindsey Compton from the University of Birmingham. Lindsey, thank you so much for joining me today.
It's a pleasure to be here.
Thank you. It's always great to talk to you. And I think your work is really, really inspiring. And I think it will capture people's imagination quite a bit, as well as to the power of genetics to really change and shape the worlds. But before we get into that, can you just tell us a bit about your background and how you got into genetics in the first place?
Yeah, so I've always been a scientist at heart. So I did an undergraduate degree at University of Birmingham in biological sciences, and took all the genetics modules available to me. I became particularly interested in genetics, just really fascinated with this idea that there is a code that underpins organisms' characteristics, and that minor changes like swapping one letter out for another in that code can cause quite massive changes in terms of characteristics. And I became fascinated with the molecular details of that. What's the rule book for life, really? So I did my undergraduate, probably enjoyed being at university far too much, so I wanted to just stay. I went straight on to doing a PhD in Birmingham, which was much easier back then to get a PhD than it is these days. So my personal tutor at university invited me to stay on and do a PhD with him, which I did. So I began to specialise more in genetics during my PhD. So that was focused on developing statistical methods for making that link between the genome, the DNA, and the observable traits or phenotypes of organisms. So it was very much a crash course in statistics and computer programming. It's very much a computational project. So that's how I got into genetics. And then actually after my PhD, I then went into teaching.
What did you do your PhD on specifically?
So I did my PhD on building methods for mapping quantitative traits. So quantitative trait locus, or QTL mapping, was the topic. So I was working with potato, which, unlike species like ourselves, is actually tetraploid. So it has 4 copies of each chromosome rather than 2. So that makes the genetics more complicated. So we were developing methods for the QTL analysis, which statistical methods that actually link genetic variation in the genome sequence with variation in traits. So I wrote some code to actually design a QTL mapping method that could accommodate the complexities that happen when you have 4 copies of each chromosome rather than 2. It just creates a lot more possibilities for the genotype of organisms and makes that relationship between genotype and phenotype more complicated. So it's very much grounded in bioinformatics type work.
There's already so many things there that I'd love to ask you about, which is terrific. But before I forget, I've raised this with the Genetic Society before. Sometimes I wonder about young people's education. They're not being exposed to genetics enough. Because most students, to be honest, that I come across who are doing biology at their A-levels, they're heading towards medicine, and that's sort of what they want to do. So at what age did you really get exposed to genetics? Was it when you were in high school, or did you come across it sort of in science communication or museums?
For me, it was A-level biology. So after school, I did A-levels, and in that biology A-level, there was a fair bit of genetics, but it just captured my interest. So I knew that I wanted to study genetics more at that point, and that's why I went to do biological sciences at university. Tough decision versus the other sciences, but for me, wanting to know more about genetics sparked that. So, like you said, there may not be that much a thorough knowledge of genetics at that level. And there tends to be a focus on human health and disease. And I think that continues as well. And it really shapes what people want to do. So our undergraduates and postgraduates who join us to do bioinformatics, as an example, often come in with very much this idea that I'm interested in cancer and human
Yeah.
health and disease, and not so much in the really broad applications of genetics, particularly plant science. And I think that bias happens very early, and it's something that work is being done to try to make the curriculum more engaging and have more genetics in it from school onwards. But it's an ongoing thing that we face.
Yeah, and at what point did plants really take over for you then? Because when I saw that plants were polyploidy and that you're dealing with not 2 chromosomes but 4 and sometimes more, I ran a mile. I was absolutely terrified. That was very intimidating. So, what was it that really switched you onto plants?
I'd like to say that I was absolutely fascinated by plants and I used to grow my own crops and everything, but honestly, it isn't the case. In my career, I think personal connection is what has really driven the way that I went. It always has been. So many examples. So when I finished my undergraduate degree, I was just still interested in everything at that point. I have to say, plant science had not particularly captured my attention during my undergraduate degree at all. So I can't say I'm a crazy plant person at all. But I just, through the connection that I had with the staff on the course, particularly my personal tutor, I wanted to carry on working in the team that I was working in. It happened that they were working on plant science, and so I got into plant related genetics, but it wasn't really intentionally. I'm very much choose people rather than projects.
Mm. A personal thing that I love about meeting scientists in the life sciences who are doing different things from me is their particular passion for their model organisms. So, I have one of my best friends is just obsessed with microbes. He's like, Sam, don't waste your time with humans. They're boring. Get into microbes. I have a friend who's deeply passionate about mosquitoes, who are a model organism, and I'll never understand that one in particular. But when I first met you, you were so enthusiastic about the potential of potatoes and what you could do with it, and it really left a mark. I was quite amazed by it. So, could you tell us a little bit about what it is to work with potatoes as a scientist?
Yeah, so as I mentioned, I didn't particularly want to go and work on plants, but once I choose to get my teeth into something, I do get very excited about it. And actually, I realised when I started my PhD and since then, just how exciting plant science is. Coming back to the point we were talking about earlier, really, that actually a lot of students don't realise that until sometimes it's too late, because at school you're not really exposed to how exciting plant science really is. But I was lucky to get into it through my PhD. I think plant science is a very exciting area to be in, particularly in the UK. The UK is really at the forefront of crop genetics and crop breeding, particularly with a few years ago there was a Precision Breeding Act, which has enabled the use of modern technologies for breeding better crops, that was being approved in England and Wales. And so that's opened up a lot of doors for really exploiting the potential of genetics to improve plants and improve crops. So it's an exciting time because many people, obviously, human health and disease, coming back to that again, is really, really important. We want less people to suffer from cancer and to die of cancer every year, but really having enough food to feed the human population, enough safe and healthy food, is really the biggest challenge that we're facing as a species. And if we don't address this, then we really are in big trouble. So I remember when I came to give the talk in schools that you invited me to, I showed a slide where there was something like 9 billion deaths globally from cancer every year. And it's a shocking statistic, and it's something that matters to everybody. But not many people realise that the number is much greater for the people who actually die of starvation every year. So we really need to prioritise research in plants so that we can actually grow enough food to stay alive as a species. It really is. It sounds dramatic, but that's the situation we're in at the moment, especially with climate change, because the crops that we've been growing for many years, whether it be potato or other crops, may not be well adapted to how quickly the climate is changing as well. So the time is really now to start applying the things that we know about genetics to breed better crops.
Yeah, your work is so amazing, and it's so easy for people to see the potential and the scope of application for what you do. And a lot of what you've just said, the general public won't really be aware of. And that's often the case with genetics. The general public doesn't understand just how far along things are. Um, how many different types of potatoes are there, and why are potatoes particularly good?
There are many thousands of different varieties of potatoes. So we tend to see in the supermarket, you see the same odd varieties, maybe 4 or 5 varieties available, like Maris Piper and other names that people might have seen. But that's just a really small set of varieties that are sold in the supermarket. There's many varieties, particularly in countries like Peru, which is the center of origin of the species, and they don't necessarily look anything like the potatoes that we're familiar with. So genetically and phenotypically, they're really, really diverse, different colors, shapes and sizes. You know, you put them all together into a picture, it's beautiful. The diversity of different potato varieties which have different properties. They taste different, look different, they cook differently, everything. But it's the same with many crops, that we just have a very narrow set of varieties that are available to consumers. But actually there's many more varieties out there. So working with potato as a crop, why is it important? It is one of the most important food crops globally. And one of the reasons for that is because, unlike crops like rice, it's really nutritionally dense. So there's a lot of vitamins and minerals in potato, and you can survive just eating potato for a very long time compared to eating rice, because it's got a lot more nutritional value, and it can actually be grown in a lot of different environments as well. It's quite adaptable as a crop. So it's been described as a climate smart option, or really a crop for the future. So we should be investing more in actually making the use of the diversity of different varieties available, and breeding better varieties that will adapt to what we have with climate change.
You mentioned Peru there, and your work has had quite a lot of crossover with South America. Could you tell us a bit about what that looks like and how that works?
Yeah, so I had a PhD student work with me recently who was actually from Peru and was very interested in working on potatoes. So one of the things we did was actually to look at what we normally work with. Actually is a European panel of varieties, which are nothing like the varieties from Peru. So the pool that you have is quite different. So through the connection with her and through colleagues in Peru, we worked with some varieties that were very drought tolerant. As an example of a trait that we want to build into potato, is to enable it to be more drought tolerant, particularly because potato has very shallow root systems, so it can be really susceptible to drought, and that can massively reduce the yield. So we wanted to understand how those varieties are so resistant. So we did some sequencing work to try to understand how the genes are expressed in those varieties, comparing one that's really drought tolerant and one that's not. And I identified different candidate genes that can actually explain the drought tolerance of those varieties. And I think that kind of work is quite important, because we often work with a small panel of varieties that are UK or European, but we don't capture the global genetic diversity. So that's what we were trying to do in that project, is to understand how the traits are controlled in those varieties that are tolerant to the stress, and how can we bring those characteristics into the varieties that we use in the UK or Europe.
Yes, and I remember seeing the picture of all the different types of potatoes, particularly those in South America, and thinking that looks nothing like a potato that I know.
Yeah.
There is an extraordinary amount of diversity there. So, how can a geneticist like yourself harvest this diversity to really solve something like world hunger? What are the tools of the trade, and what are the potential applications?
Yeah, so always what we're trying to do is to find out what is the genetic change that confers the useful trait, like drought tolerance or resistance to a particular disease. And it's never going to be just one genetic change, but if we can identify something that has high importance towards the drought tolerance or other traits, we can then actually introduce that trait or that genetic change into the varieties that we use in the UK. So there's different ways that we can do that. So we can use traditional breeding, or we can use more modern methods of gene editing, which, as I alluded to earlier, the legal act that actually allows us to use gene editing approaches in the UK was approved and passed in Parliament in 2023, which puts us in a really exciting place in terms of plant research in the UK, because we can use precision technologies to actually change the genetic profile of a potato variety and see if it has the intended effect on the phenotype. So if we know what the change is in terms of the sequence, we can edit the genome so it has that sequence and then test it in different environments and see if it does confer an improvement in the trait. So the advantage of the precision breeding or gene editing, it's the same thing really, is that it's much, much faster than crossing together different varieties and raising the offspring. It can take 10 years to create a new variety of potato or other crops by using crosses. But actually with the gene editing technologies, it can be done quite quickly in the lab. So I haven't actually done any gene editing with myself, but obviously it's something that I keep abreast of because it's very relevant to my own research.
Yes, a lot of people have heard of CRISPR.
Yeah.
The Nobel Prize awarded for it 2020, I think. And you also have prime editing and base editing now as well. Incredible technologies. And you mentioned there that can accelerate the process rather than just so are we talking about sort of hybrid potatoes taking the best of what's in one part of the world or one species and sort of crossing it with another to sort of make something that's bigger, better, more nutritious?
Yeah, exactly that. So you're trying to identify regions of the genome that can further advantageous trait or boost that trait in some way and to introduce it. And they're very much the quickest way to do that is to use some form of gene editing. And like I said, I haven't actually done gene editing in the lab myself, but the technologies to actually do it keep advancing really, really quickly. And it's a challenge as a scientist to keep pace with exactly the different technologies and the different methods. But what they all have in common is that they're precisely introducing a change, and it, by definition, it should be a change that could have happened naturally. It's just that, as we were saying at the beginning, we don't have the time to wait. You know, as a species, we need the better crops and we needed them years ago. We need them today. Okay. So we can't wait 10 years for that change to happen naturally. We needed a way to actually make that change happen in the lab. And now it's possible. There's lots of different examples that different plant breeding institutes and universities have done to create varieties and new varieties. So it's basically the same variety, but just boosted by making one or more genetic changes. So some of those changes might improve the nutritional profile of the crop. So tomatoes have been made that produce more vitamin D, as an example, to make them better quality. Some changes are in terms of the bigger food security picture isn't about just having more food, but actually reducing food wastage is a major problem. So bananas have been genetically edited so that they don't turn brown. So a huge amount of waste is generated through that pathway. And similarly, food has to be safe, of course, as well as nutritionally high quality. So a recent example of that is where bread wheat has been engineered to have less asparagine, which is an amino acid in the grains. And that's important because during cooking, it can lead to acrylamide formation, which is toxic. So by genetically engineering the wheat, they just have slightly less of this amino acid. It's not having any detrimental effects on any other traits, but it's reducing toxic compounds when you cook the crop. So there's loads of different examples like that of where gene editing has been applied to produce a better quality or safer crop.
That's amazing. That's absolutely extraordinary. And again, most of the public won't have heard of that. They might have heard of golden rice. I suppose golden
Yes.
rice is the really famous one.
Yeah, absolutely.
And yeah, a Nobel Prize has been awarded for this in the 20th century for just the increasing the productivity. But golden rice is who knows how many lives that's saved.
Yeah.
Now you mentioned climate being a big challenge here to crop production. And when you're trying to enhance potatoes so that they are more drought resistant, more heat resistant, more resistant to pests? Are you occasionally needing to take genes or genomic information from other species or sort of other organisms?
Yeah. So often that is the case. Yeah. So um, in the example of potatoes, because we know that there's a huge diversity in Peru as an example, and they're going to be grown in very different conditions to the conditions that we grow them in the UK. So actually understanding what makes, if you already have some of those crops growing at higher temperatures, and you can find out what leads to that tolerance trait, then you can introduce that to varieties which are typically grown at lower temperatures. It's the same with forest trees as well, as another example, not just crops. So there's a lot of reforestation and new woodlands being planted in the UK to try and combat climate change. But it's not good to just plant the same varieties that we've always had historically. So one approach would be to actually look at countries further south where the trees are actually adapted to higher temperatures and bring that kind of genetic varieties to the UK, because then the tree that you plant today will actually be able to survive the conditions that it will be in 10, 20, 30 years time. So there's plenty of examples of that where you use existing material from different places that already have some of the traits that you're interested in.
That's fantastic. Potatoes are obviously very, very hardy. They've been around for
Yeah.
hundreds and hundreds of millions of years, I'm sure. And one of my favorite films and books is The Martian, where Matt Damon grows potatoes on Mars to help him survive. In real world applications, we've seen people grow Arabidopsis in lunar soil, lunar regolith. Why potatoes? Potatoes have got to be one of those top five, top 10 food groups. But what is it about potatoes in particular that is so helpful for us?
I'd say that the adaptability of the crop. So already, even without breeding more varieties, it can be grown in much more diverse range of conditions. So temperature, amount of water, all the different factors that affect plant growth compared to other crops like rice. It is much more highly adaptable. So it can be grown in a much more diverse range of environments. So in that sense, with climate change not just being a directional change, but it's more, there's greater fluctuation in conditions than ever before, then it's probably one of the best options for being a climate smart crop, even without the genetic work that we're talking about.
Mm.
Not to mention, as we talked about before, it's just very nutritionally dense as a crop. So for a small amount of food, you just get more nutrition, which is obviously good in terms of feeding the population and having enough nutrients to be healthy.
Yeah, terrific. I don't know how we'd do without chips as well. Certainly not in Britain or in the West. Um, but you mentioned earlier computational methods and bioinformatics, sort of the marriage of biology and computer science, being part of your job. Could you explain some of that and what that looks like and how you can harness that for your work?
Yes. So my job is partly research, but also a large part teaching as well. So we have a master's program in bioinformatics at the University of Birmingham that I lead. And that really, we always like to describe bioinformatics as being data science for the life sciences, and that might sound incredibly broad, but that's because it literally is. So you're applying data science techniques of analysing data to any kind of biology related data set. So the types of problems that you can be tackling really, really, really broad, ranging from the health of forest trees through to human cancers. And so on that program, we try to expose students to the full power of bioinformatics to address different things, whether it be a human cancer and understanding the evolution of tumours, through to actually the types of plant science research that we've been talking about, or other environmental research, research in psychology, lots of disciplines that interact with bioinformatics. So it's really about making sure people have the skills to do computer programming on one hand, but also to understand the different types of analysis that you can apply to data and choose the right kind of analysis to pull something out that's biologically meaningful. Because as you probably already know, the bottleneck in modern biosciences is not so much about generating the data, but about actually having the people who have the right skills to analyze that data, to know what to do with it and be able to make meaningful conclusions from that data. So that's what the bioinformatics is all about, is building the next generation of skilled scientists who can analyze the exploding amounts of data in biology. So it's an exciting program to run, an exciting field to work in. If you look at, I saw a chart the other day which was plotting the number of published research papers that are related to bioinformatics, and it literally just goes up and up and up every year over the past 20 years. So we always said it's a rapidly growing field, and it feels a bit old now, but we keep saying it because it literally is true. But that bioinformatics is really, we think of it as the future of biosciences, because even if you would prefer as a scientist to stay in the wet lab and not do the data analysis and the bioinformatics side of things, increasingly that's becoming more difficult to stay with doing that without engaging in the bioinformatics. So it's an exciting area to be part of and to teach as well.
Yeah, it's a great career to be getting into for young people and for students. And you're right, there's certainly no shortage of need there at all. How does sort of model building or the data analysis side of things inform sort of potential therapeutic targets or sites of interest? What does that look like?
So in a project for, let's say, trying to understand the genetic differences between tumour tissue and healthy tissues, that's quite a common type of project that we have people offer to our bioinformatics students, where they're looking at, say, breast cancer or a different type of cancer, and they compare the genetic profile of normal tissue to diseased tissue to try to understand where the differences are. So what's happening within the tumour in terms of the way that genes are expressed. For example, that's different to the healthy tissue. So lots of different levels of biology can be targeted. So you can look at things at the DNA level, or you can look at things at the transcriptome level and look at the expression of genes genome-wide. You can also look at epigenetics as well. So commonly the methylation profile of the genes across the genome in the tumour versus healthy tissue. And there are other levels as well that we can look at, like metabolomics as well. So how does the profile of metabolites, small molecules inside the different tissues, differ? And similarly, we can do proteomics to focus on proteins. What's the different proteins that are present in a tissue and how abundant they are in different types of tissue? So in bioinformatics, you can look at things on many different levels. And you'd be applying types of data analysis that are specific to that type of data. Or you can, rather than focus on one level, you can collect data across multiple levels. Then you have the challenge of integrating the information that you get from the gene expression profile, the protein profile, the metabolite profile into more of a holistic story as to what's going on. So the data sets can become quite big and quite complicated, but essentially, if you can identify something that's dysregulated in the tumour condition versus normal, for example, consistently in a cohort of patients, then you can potentially develop treatments that would target that particular gene and the way it's expressed, as an example, as a form of targeted treatment. And different people in the population may have different underlying mechanisms in their cancer, so you can personalize the treatment according to the genetic profile of that individual. And I do think that's important. It's not my research field, but I have sort of stayed abreast of the literature, and I know there's examples where if a method, it's important to know the genetic profile of individuals in order to tailor the treatment. So a lot of methods have been developed from white populations as an example, and different global populations and ethnic minorities are really underrepresented in general in this kind of human health and disease research. And although that's changing, I have seen examples where if you take a treatment that's developed based on genetic profile of white people and then you apply it to a population with a very different genetic profile, it doesn't necessarily not work. It can actually have detrimental impact as well. So it can actually lead to worse health, which is opposite to what you're trying to achieve. So there's a lot of exciting things that can be done, but there's also equality, diversity and inclusion issues as well in human genetic research in terms of what populations get prioritized for understanding the genetic basis of disease and making sure that there's a understanding that you can't just apply a solution that's developed with one kind of population geographically and apply it to everyone as well.
Mm. Yeah, that's amazing. There's so much there we could get into. I'm so excited about how bioinformatics can shape the future. And I encourage students as well towards your master's program, because people can get into bioinformatics from a biology background or a computer science background. So
Yes.
it's it's an exciting world. I'd love to give you the floor here to paint a vision of the future in terms of how genetics can change world hunger and starvation. What does that look like in terms of what are the best possibilities and what are you really hopeful for?
Yeah, so I think the best possibility of using genetics to breed better varieties of crops that will be useful in our rapidly changing climate to feed the future growing population. The best approaches are a combination of the classic ways of understanding what genetic changes linked to what traits. So we can do QTL mapping approaches that I mentioned earlier, or genome-wide association studies where you take a big panel of genetic varieties, like a panel of 300 potato varieties that we work with. You collect data on the phenotypes that you're interested in, and then we use statistical methods to bridge the genotype and phenotype and find out what are the genetic changes that underpin the traits. So I think we can use the classic methods to find the genetic variants that we're interested in. But I think when it comes to plant breeding, the future has to embrace gene editing approaches because, as we were saying earlier, classic breeding and crossing together varieties and selecting the ones that you want from the offspring is intensive, it's laborious, it takes too long. And if we really want to address the need to create the better varieties sooner, we have to embrace the modern technologies. So I think the way forward is for scientists in the UK to make the most of being in a good position where there's already been an act, the Precision Breeding Act, supporting the use of these technologies. We have to make the most of that in the coming years to stay at the forefront of genetics and genomic research in plants and actually produce those varieties in good time, really.
It's very exciting. Genetic engineering is we're in a CRISPR world now. Is how I
Yeah.
put it to people. Everything before 2011 is a very different world now.
Yeah, I think
And
there's a parallel, isn't there, with generative artificial intelligence, and there's still people who want to resist using it or resist embracing it. For example, in our teaching, we can say it's banned and no one's allowed to use it, but in reality, we can't just naively do that. In reality, people can and will be using it, and we have to embrace it. We have to make use of it rather than be scared of it. And I think the parallel is with the genetic technologies as well, that is the world that we're living in, and we have to embrace it to the extent that we can and not avoid what is very complicated. So the Genetic Technology Act, a Precision Breeding Act as an example, there's a lot of complexity in terms of the way it needs to be regulated, the way it needs to be implemented so that it benefits society and it doesn't benefit some groups and not others. There's a lot of complexity in secondary legislation that will need to come into to play to support that, but we mustn't shy away from that. We must actually embrace those technologies and put in place the things that are needed to get the most out of it. And it's the same with Gen AI, I think.
Yeah, I completely agree. And I think there's a snowball effect here, which makes the biological sciences so exciting right now because AI has brought along things like AlphaFold, obviously bioinformatics and the marriage of computer science and the life sciences has brought about this data revolution, which has just given us so much more information to work with. But now we have the tools as well that have come along together in things like AlphaFold, which the Nobel Prize was awarded for last December,
Yeah.
and things like CRISPR and now base editing and prime editing. You have so much more information and you have so much more powerful tools, and they've all come at the same time,
Yeah.
which has brought about, I think, the biggest revolution in biology since maybe the 1950s. Um, so it's a very exciting time, and we can think about what the future can be, but in many ways, it's as big as our imagination.
Yeah.
I'm very
Absolutely.
glad we're in the game and ahead of the game here in the UK.
Yeah. Absolutely. It's a very exciting time.
I'd like to ask you as well about the Genetics Society. So the Genetics Society here in the UK, it's got a journal associated with it called Heredity, which is a fantastic journal. And you've been on the board. So could you tell us a little bit about the Genetics Society, what it does, and why it's great?
Yeah, absolutely. So, yeah, I have been on the committee of Genetics Society for some years. The society attempts to bring together the community of geneticists in the UK and support that community in the ways that it can. So, we have a couple of conferences each year in the spring and the autumn on different topics, very broadly related to the broad applications of genetics that we've been talking about. We also have grants that people can apply for example, fieldwork grants to support students to come and work with academics in summer, for example, to do an 8-week summer research project, fully funded. We have different workshops as well to support students, for example, in terms of communicating their science, which is really important. Skill as well, being able to do the science itself. So we have things like that we encourage people to get involved in at all levels. And the Society's journal as well, Heredity, also publishes up-to-date research in genetics and really supports younger researchers as well, which is nice. So whenever we have conferences in Genetics Society in general, we do have a lot of support for early career researchers, but the Genetics Society journal Heredity, we also have prizes like, for example, the best student paper of the year and things like that really encourage the participation of the next generation and recognition of what people have achieved as well, and various prizes to recognise achievement at different levels as well. So for one of them, for example, is the Kenneth Mather Memorial Prize in quantitative genetics. So there's lots of different prizes that people can nominate their students for to get people the reward and recognition. So all of that is done through the Genetics Society. So it's really good to be involved in it. And as a student, membership is very cheap as well. Off the top of my head, it's only £5 or £10 a year, and you get a quarterly Genetics Society magazine as well, which is really engaging, accessible content. And I think, from my perspective, one of the hardest things is always keeping up to date with how fast things change and the research that people are doing, even within your own field, let alone knowing what's going on elsewhere. So Genetics Society magazine is quite nice for that, because you can get an idea of really the amazing diversity of things that people are working on. So I can zoom out and stop thinking about potatoes and think about the patterning of the colours on butterfly wings or whatever it is that people are working on. It's really fascinating. So for me, the Genetics Society provides that kind of continuing professional development as well, so you don't just stay narrowly focused on your own field.
Yes, it's so encouraging, and the conferences and the gatherings are a
Yeah.
great place to network and meet other people who are involved in areas close to or that can cross over with your research. And I just always found it tremendously encouraging as a
Yeah.
student as well. And the conference this year, I encourage people to check it out. We have Svante Pablo speaking, who won the Nobel Prize for sequencing the Neanderthal genomes. That's going to be very, very interesting. And it's
Yeah.
it's a fantastic way to help your career, but also stay in touch with what's going on everywhere. Um, what advice would you give to a young Lindsey Compton or students out there who are looking to get into genetics? What sort of routes should they go? What should they look out for? What should they study at school? How can people get into genetics and plant genetics in particular?
Um, it's a really good question. Let me think about this one. So I think the most important thing, and I wish I'd have known this when I was younger, is just to talk to as many people as possible. So I was too terribly painfully shy to do that as a youngster. And even now I find conferences quite intimidating. But I think the more you can try to do that from a young age, because that's how you find out what opportunities are available. So know the things that I've got involved in, and not normally necessarily things I've ever actively pursued, but it's the people you know who say, hey, come and do this. That's, I think, building your professional network earlier in genetics or any other field, I think that would be my biggest advice, and to get out there and attend events like the Genetic Society conferences or smaller workshops and symposiums, just to get to know what people are working on and build your professional network, really, because I think that's how doors open usually, rather than, for me at least, that's how it's happened in my life. Rather than me searching for an opportunity out of the blue, things have come about because of someone I happen to know. So that would probably be my best advice, is to I would like to force my earlier very shy self out of her comfort zone into the community of scientists.
Yeah, that's great advice, definitely. And for students who are watching and following this as well, just stay curious and keep in touch
Yes.
with the sciences. I think in genetics as well, like don't panic if you're not amazing at maths. There's still a place for you in genetics. There's still a place for you in science. But stay curious and stay interested. Thanks so much for your time today, Lindsey. It's always a pleasure. And if people want to find out more about you in particular and your research on potatoes, where could they go?
Probably looking at my LinkedIn profile would be the best place to start.
Cool. Terrific. Thank you. Look forward to seeing you at the conference if you're there. And
Yeah.
thanks so much for your time today.
Thank you. Great to chat with you about science. Thank you.
Thank you so much for watching. Don't forget to like and subscribe. Stay tuned for more amazing conversations with incredible researchers who are changing the world we live in and driving humanity forward. Feel free to check out my Instagram at polymath_Sam, or go to my website at www.sam-mckee.co.uk.